morning, everyone. My name is Monica, and today I will be presenting along with my teammates on the fascinating topic of biomanufacturing, 3D bioprinting, and organ on chip technology. Our guiding theme is microfluidics, the central technology that connects all three. Why focus on microfluidics? Because it allows us to handle fluids at microscopic scales, creating an unprecedented level of precision in biotechnology. This precision is essential for developing new medicines, testing their safety, and even engineering tissues and organs. In this presentation, I'll first introduce microfluidics as the glue that unifies these fields. Then I'll explain each application, that is, organ on chip, biomanufacturing, and 3D bioprinting. Finally, I'll show how all three coverage into an integrated future which is already starting to reshape biomedical sciences and healthcare. So let us begin by understanding what microfluidics actually is. Microfluidics is the science and technology of manipulating very small amounts of fluids, often just a few microliters or even nanoliters inside channels that are often no wider than a human hair. Why is this so powerful? Because in biology, the environment matters. Cells behave differently when they are in static, flat petri dishes compared to when they are exposed to real life conditions such as fluid shear, chemical gradient, or dynamic nutrient exchange. Microfluidics allow us to create these in vivo conditions in vitro. That means we can mimic blood flow, nutrient transport, or even immune cell migration on a tiny chip. The benefits include unprecedented control. It allows exact regulation of flows, mixing, and gradient. Physiological relevance. Models built with microfluidics behave more like human tissues, improving the accuracy of experiments. Scalability. Microfluidic system can be miniaturized, parallelized, and automated, which is ideal for both research and industrial bioprocesses. To give a simple example, imagine mixing two liquids by hand in a beaker. The process is random and slow, but in a microfluidic chip, mixing can happen within milliseconds, producing perfectly uniform solutions. This level of control is what microfluidics, the backbone of modern biotech. Let's move to our first application, organ on chip technology. Organ on chip devices are essentially microfluidic systems designed to mimic the structure and function of human organs. They contain living cells arranged in a way that recreates tissue-tissue interfaces surrounded by channels where fluids can flow just like blood would inside the body. Why is this a big deal? Traditionally, drug testing has relied on 2D cell cultures or animal models, but flat cell cultures oversimplify biology and animals do not often accurately represent human physiology. That is why so many drugs fail in clinical trials despite looking promising in earlier stages. Organ on chip fills this gap by providing human relevant, physiologically accurate models. For example, a lung on chip can recreate the expansion and contraction of breathing, allowing researchers to test how inhaled drugs or pollutants affect the lung. The heart on chip can stimulate the beating of cardiac tissues and be used to detect side effects of new medicines on heart rhythm. Gut on chip can reproduce the digestion process and even host microbiome interactions. These platforms are already being used for drug efficacy testing, ADME tox studies, which stand for absorption, distribution, metabolism, excretion, and toxicity, and even for studying complex diseases like cancer or neurodegenerative disorders. Now, how microfluidics make organ on chip possible? Firstly, it enables physiological stress. In the human body, endothelial cells in blood vessels are constantly exposed to flowing blood. Without this stress, their behavior changes. Microfluidic channel can reproduce this flow, keeping the cells in a more natural state. Secondly, microfluidics allow on-chip gradient generation. Many biological processes, such as immune cell migration or wound healing, are guided by chemical gradients. By controlling fluid flows, we can establish precise and stable gradients inside a chip, something that is very difficult to achieve with traditional methods. Thirdly, microfluidics supports multi organ coupling. This means that several organ on chip models can be connected together using microvascular networks and recirculating fluids, stimulating how organs interact in the human body. This concept called body on chip brings us closer to a complete in vitro human models for drug testing. Latest developments. So, recent years have been seen incredible advances in organ on chip. One major breakthrough is the development of body on chip systems. Researchers are working on platforms that connect up to 18 different organ models, including vascular and excretory systems. These platforms are expected to revolutionize drug testing by offering far more reliable predictions than animal bodies. Regulatory policy is also supporting this shift. The FDA Modernization Act 2.0 encourages the use of microphysiological systems and new approach methodologies in drug testing. This represents a clear movement away from animal testing with microfluidic system at the center of this transition. The FDA is also establishing performance standards and validation work streams to ensure organ on chip models can be used confidently in regulatory submissions. So, we are at the turning point. The technology is maturing and the regulatory environment is ready to embrace it. Now, SK Monica Devi will continue. Gaps. Of course, organ on chip still faces challenges. Standardization. Different labs uses different chip designs and protocols, making result harder to compare. Reproducibility. Slight variation in chip fabrication or cell culture can lead to inconsistent outcome. Data sharing. The complexity of organ on chip data requires better framework for sharing and interoperability. Manufacturing scale out. Producing these chips in large quantities at low cost remains difficult. Validation. For regulatory approval, universally accepted validation frameworks are urgently needed. Addressing these issues will be key to scaling up organ on chip from research labs into industry and clinical practices. Now let us turn to biomanufacturing. Biomanufacturing is the use of living systems such as bacteria, yeast, or mammalian cells to produce variable biological products. These include therapeutic proteins, vaccines, cell therapies, and gene therapies. Traditionally, these products were made in batch processes, but batch processes are often slow, expensive, and variable in quantity. The future lies in continuous biomanufacturing, which is faster, more reliable, and easier to scale. But microfluidics plays a central role here because it allows precise control over the reaction environment. At small scale, microfluidic reactions can maintain consistent conditions such as temperature, pH, mixing, which directly translates into higher product quality and reproducibility. This is especially critical for next generation products such as cell therapies and mRNA vaccines. Moving into biomanufacturing microfluidics tools. A great example is the mRNA vaccine resolution. During the COVID 19 pandemic, we witnessed how quickly mRNA vaccines can be designed. But to deliver mRNA safely into the body, it must be packaged in lipid nanoparticle or LNPs. Microfluidic mixtures enables extremely rapid and uniform mixing of mRNA with lipids, producing nanoparticles that are highly consistent in size and encapsulation efficiency. This level of control is essential because even small variation can affect vaccine stability and efficiencies. Microfluidic systems are also scalable. By parallelizing chip production can be scaled from milliliters to liter per hour without losing control over particle size and quality. New breakthrough includes 3D printed microfluidic mixers, which have been shown to increase throughput by 5 times while maintaining precision. This means vaccines and biologies can be manufactured more quickly, reliably, and at low cost. Next will be carried out by Monisha.
About 3D bioprinting. 3D bioprinting. This technology uses printers that deposit bioinks, which are mixtures of living cells and biomaterials, to build three-dimensional tissues layer by layer. There are multiple methods: extrusion based bioprinting, where continuous strands of bioink are extruded. Inkjet bioprinting, which sprays tiny droplets of cells. Light-based methods such as digital light processing, DLP, and stereolithography SLE, which use light to cure bioinks into precise patterns. Emerging methods like fresh printing allow delicate structures such as vascular networks to be printed with high resolution. The applications are immense. Regenerative medicine, building tissues, or even whole organs to replace damaged ones. Drug discovery, printing disease models to test therapies. Personalized medicine for creating patient specific tissues for customized treatment. Microfluidics complements bioprinting by providing perfusion channels that keep the printed tissues alive and the functional solving challenges of the nutrient delivery. Now, let us see the convergence integrated future. Individually, organ on chip biomanufacturing and 3D bioprinting are powerful, but their greatest potential lies in their convergence. Bioprinted tissues can be integrated onto microfluidic chips, creating advanced experimental models that are both complex and physiological accurate. Organ on a chip platforms can serve as a release test through biomanufacturing and offering more reliable data than animal models. Closed loop microfactories are on the horizon, where drug synthesis, testing, validation happen seamlessly within one automated system. This vision of integration accelerates discovery, ensures quality, and reduces cost, while in minimizing reliance on the animals and maximizing human driven. Now, coming to the conclusion, to conclude, microfluidics is not just another tool in biotechnology. It is the foundation that connects and empowers the most transformative technologies of our time. In organochip, it provides human relevant testing platform. In biomanufacturing, it ensures precise and scalable production of vaccines and biologics. In 3D bioprinting, it enables the creation of functional living tissue. And in future, by bringing all three together, microfluidics will help us realize the vision of fully automated and integrated microfactories for medicine and research. And future of the biological engineering is integrated, precise, and microfluidics is driven. Thank you for your attention.